This is a research presentation entitled Concetta Antico's Potential Tetrachromacy, done by Kimberly A. Jameson and Alissa D. Winkler on Friday, August 15, 2014. The sun's radiation illuminates the earth. It does so in a variety of environments and across many different ambient lighting circumstances. It also does so for a wide range of visual processing modes. The portion of the sun's energy that is actually visible to humans is just a small sample of the sun's radiation. The humanly visible range spans roughly 400 nanometers on the short wavelength end to 700 nanometers on the long wavelength end. Beyond 400 nanometers, ultraviolet light wavelengths occur, and on the other end, beyond 700 nanometers, are the infrared wavelengths. While sunlight, or light in general, is essential for perceiving color in real-world objects, it does not actually determine the colors that are seen in the world. This is because color depends entirely on an observer's visual system. We know this because we have knowledge about the kinds of cells that exist in human retinas. We know that normal human color vision is typically experienced through three different kinds of daylight sensing cells called photoreceptors, or cones, as shown here. These photoreceptors number in the millions and are located in a photosensitive layer in the back of the retina, and they respond to specific wavelengths of light as shown by these three curves. Individuals who inherit the genes for all three types of photopigment classes have normal human color vision. However, some individuals may not inherit the genes or may have a deficiency in the long wavelength sensitive, or the medium wavelength sensitive, or the short wavelength sensitive cone class, and this will affect their perception of everyday objects. So a normal trichromat will see this field of tulips in the following way, and we can simulate how an individual who is missing the long wavelength sensitive class will see this field, how an individual who is missing the medium wavelength class will see this field of tulips, and how an individual missing the short wavelength class will see these tulips. You might ask, how does the perception of color depend on these photopigment types that an observer has? Well, here you see a physical measurement of a specific patch of color with wavelength and intensity. But can we tell from this curve alone what the color of this particular patch will be? The way a trichromat observer encodes the spectrum of light depends on their cone classes, and it will typically be seen by a trichromat observer as a bright shade of red. This is because a normal trichromat observer generally possesses these three sets of photoreceptor classes: long wavelength sensitive, medium wavelength sensitive, and short wavelength sensitive. And these three types of photoreceptor classes define what a trichromat observer is, tri being three and chromat being color, for the three cone classes that peak in different regions of the spectrum. By comparison, there are some individuals who have the potential for tetrachromacy. A tetrachromat observer is an individual who has an additional photoreceptor class in addition to the usual three cell types that a trichromat has. So this tetrachromat observer also has a receptor class intermediate to the medium and the long wavelength sensitive class that normal trichromats have. And this is referred to in this talk as an L' photoreceptor class, shown by this dotted curve. The interesting question is whether or not this extra class of cells implies something different for the perception of color for tetrachromat observers. That is, we don't know whether it will alter the shade of red that we saw previously to be a lighter shade, or whether it will produce the sensation of a lighter red with a yellowish component in it. It could also introduce some other quality, like shimmer or gloss, to the color patch. The answers to these questions remain very uncertain, but everything we know about nature tells us it should make a difference in the human visual processing of color to have an extra photoreceptor class in this region of the spectrum. Some examples from nature help us understand how this may be the case. A wide range of visual processing systems exist across the Earth's inhabitants, and there are many cases of tetrachromacy present in nature. One example is shown here with the European starling. European starling is a bird that has an extra photopigment class that is sensitive to the wavelengths in the near UV region of the spectrum, outside the visible range of humans. Having this extra photopigment class allows these birds to have an extra dimension of color experience that humans don't have. Here is an example of a colorization attempt to suggest what birds and humans might experience, and you can see that the colors are very different across these two scenes. So while we can't actually know what a bird sees, in a sense we can know that extra pigments produce highly variable perceptions of colors across birds and humans. This is a common result in nature. Tetrachromacy is demonstrated among several species of birds, fish, amphibians, reptiles, and insects. And these kinds of examples serve as an important illustration of how perceived color depends on the observer's visual system. So here, for example, you have the same orange cornflower viewed by two different organisms, the human visual system and the butterfly visual system. It's even more dramatic if you consider the bee's visual system. So in these cases, the color vision of these organisms is pointing out an important region of the flower in ways that are useful for butterfly and bee survival. The knowledge of what these other creatures see and the prospect that some humans may have an extra receptor class in their retinas has created much excitement among scientists and the general public. Naturally, from the outset, the idea of potential human tetrachromacy has gained much attention in the popular media, some of it with fantastic spin-offs of extra powers of perception bordering on the science fiction, and suggesting that tetrachromat humans may perceive on the order of 100 million colors. But this and many more questions still remain unanswered and yet to be proven by scientific study. In case you were not already aware, you may find this exciting. According to tests carried out by a leading independent laboratory conducting research on photopigment opsin genes, Concetta Antico possesses the genetic building blocks needed for perceptual tetrachromacy. She has what's called an X chromosome-linked polymorphism. Genetic tests conducted suggest that the peak sensitivity of Concetta's extra photopigment class lies between the normal trichromat L cone peak and the normal trichromat M cone peak. This means that she has a fourth pigment class with a sensitivity in between these two ranges, and it is a shifted form of the L cone. In addition to this genetic potential for tetrachromacy, Concetta has a considerable history of art training and extensive perceptual learning experience from using color at an early age. She also works daily evaluating the uses of color and light and making hundreds of color space decisions every day, whereas the typical individual will make only a very few daily decisions that involve uses of color and light and so forth. This intimate daily connection with color enriches Concetta's experience and makes it possible for her to cognitively manipulate subtle color differences that may arise from her retinal tetrachromacy in empirical tasks and empirical settings. In our design of experiments to test Concetta, we asked what some of the important considerations would be to factor into the design of the test. One consideration is the incorporation of circumstances that allow chromatic contrast and brightness contrast effects to play a role in the perception of color. Let me give you a demonstration. Here is an example of a visual illusion created by Dale Purvis and Bo Loto. As you'll notice on this Rubik cube, there are two kinds of squares that stand out. The brown square on the top of the Rubik cube and the orange square that's on the side of the Rubik cube in shadow. If I took a piece of black paper with two holes in it that exactly occur where the brown and the orange squares are, and I place them over this particular visual display, what you would see is actually the true chromaticity or the true color of these squares. And you'd be surprised because now they both appear as two brown squares instead of an orange and a brown square. What is going on here is that your visual system, which includes your brain, has learned to make inferences about color appearances in shadow, and it led you to two different subjective experiences based on its interpretation of the viewing context in which the two identical squares, or the spectra of the squares, are displayed. This is called a brightness contrast effect, and it's an important effect in understanding how the brain perceives color in the world. A second factor that we chose to include in the design of our empirical test for Concetta involved using low light adaptation levels where we suspected differences in Concetta's perception might be easier to demonstrate. The reasons for all the discussion about the tests to use to show Concetta's potential tetrachromatic difference is that most of the off-the-shelf types of tests that are out there are very inadequate for demonstrating potential tetrachromacy differences in color perception. So we knew we had to think outside the box on this one. And instead of evaluating color directly, we instead decided to look at measures that are known to be related to the collective contributions of all photoreceptor classes, namely measurements of luminance variation. Based on pilot research, we decided to try a technique for establishing equal brightness settings for different color points across color space. The method that we used is called the minimal motion phenomenon. This phenomenon works the same way motion pictures do. Frames of still images are flickered at a certain rate, and this allows for the seamless perception of motion across these different flickered stimuli. Here you have an example of a schematic stimulus. Here the illusory motion stimulus is shown for one color, namely green, in the context of a gray background. These 4 frames that are shown are presented over time at different flicker rates. And if the flickered green patch turns out to be subjectively lighter to the subject than the adaptation background, then it will, quote, line up with the light gray in the successive frames that are flickered, and illusory motion will appear to spin clockwise. By comparison, if the flickered green patch is subjectively darker than the adaptation background, then it will line up with the dark gray in successive frames, and the illusory motion will shift and appear to spin counterclockwise. This is the technique that we use to find each individual's subjective isoluminant values, and the details of the method are explained elsewhere in a technical report. The important point is that the method yields very specific types of settings that we refer to here as MM, or minimal motion isoluminant settings across color conditions. So in the experiments that we ran several subjects, but today we're just going to consider comparisons among 4 possible participants that we assessed, 4 females. The females fall into 2 groups. One set of females are trained professional artists who make a living selling their artwork. These are the subjects that are shaded on the left side of this dotted line, and they are subjects CA and TC. Subjects are represented by squares and triangles here. And if you have a subject that is a four-sided symbol, then these are potential tetrachromats. Compared with subjects who are three-sided symbols, these are potential trichromats. We can ask some interesting questions with these trichromat and potential tetrachromat artists and non-artists. For example, the first question we can ask is whether or not, independent of art training, photopigment genotype alone differentiates subjects in this task. So for example, this group of subjects may be more similar than this group here. And if this was the case, they would be differentiated by their genotype alone. The second question we can ask is, does a life history of art training by itself seem to be more important than photopigment genotype for the task that we're looking at? Another way of asking this question is perhaps instead, life history of art training is an important factor in addition to photopigment genotype for this task, in which case the potential tetrachromat who is the artist would be differentiated from the others in the experiment. These are some of the questions that we looked at with the data observations we did among these 4 female participants. And here are their preliminary results. What I'm showing you here is a curve that is a measure of performance difference between 2 subjects. The color gradient on the bottom are 20 colors that we tested in color space for this isobluminance task. And the curve represents the difference between 2 subjects' observed settings as measured in the RGB space by Euclidean distances. The details of how this is computed is not important. What is important to note is that there are significant differences among these 2 normal participants that tell us that there are regions of the color space where the subjects are not setting the same isobluminant points in the task. In the case of this graph here, the maximum obtained between these 2 normal subjects in terms of a difference is 8 Euclidean distance RGB units, as shown in the upper left-hand corner of this graph. That is compared to the average difference that is seen for the same subject repeating the experiment more than 1 time. This average variation in repeated measure performance for a single subject was observed as about 1.2 units of Euclidean distance. This 1.2 units gives us a sort of a baseline of how much variation one would expect to see between an individual running an experiment from one time to another. If we now consider Concetta's data compared with an average normal control subject, we see the following pattern. Firstly, the differences between Concetta's isobluminant settings and the average normal control settings is significantly larger than the differences seen between the 2 normal controls, obtaining a level of 14 RGB Euclidean distance units compared to the 8 shown on the previous slide. Viewed here in the context of the other 2 curves that were shown, we can appreciate exactly where these regions of differences are most substantial in Concetta's performance. The solid black line on the top traces the curve of Concetta's isobluminant setting data. You can see that in the region that involves the reddish primary in the display, or the red portions of the spectra to which the long wavelength cones are most sensitive, is the region where Concetta is showing the most substantial differences from the normal control subject. The comparison in the middle is the control subjects AW and TC and the difference seen between their isoboluminance settings. And it's clear that that is significantly lower than what's observed for Concetta compared to the normal control, as it is, as expected, different from the observation of the baseline performance between one observer's repeated measures. It's important to note that the actual result that you're seeing with Concetta's difference is linked to the subjective brightness or the luminance in Concetta's equiluminance settings. Here we see the red line tracing Concetta's measured luminance of her settings compared to a single normal subject, AW, shown in the blue line. And you can see that in the reddish region of the spectrum, those stimuli Concetta requires less luminance for in order to establish an isoboluminance setting. We tested 20 colors around the hue circle in these experiments, and the result I just showed you confirms a result that we expected to find, which is that Concetta, compared to an average trichromat control, arrived at different levels of settings for a certain region of the space that involves the long wavelength photosensitive pigments, and shows a different pattern of settings. This result suggests that Concetta is exhibiting important differences from control subjects in this task. We also want to say that the observed differences are not chromaticity based, so it's not that there's a shift in color per se. The differences are actually luminance based, which suggests the kind of role that is played by Concetta's extra photopigment class. Finally, if we compare the difference between Concetta's data and the trichromat control data with the difference between Concetta's data and the other potential tetrachromat that was tested, we notice that Concetta is even slightly more different from the other potential tetrachromat at the maximum level of Euclidean distance that she obtains in certain regions of the color space. This finding is somewhat expected given the design of our experiment and suggests a complicated relationship among these participants that will be further examined through study in the future. It is, however, consistent with our belief that perceptual learning is an important prerequisite for the realization of functional tetrachromacy. That is, comparing Concetta to our other potential tetrachromat suggests that her training as an artist has shaped her into a strong tetrachromat, whereas the other potential tetrachromat may only be a weak tetrachromat because she has lacked the artistic training from an early age that helped her develop the tetrachromat potential that she was given through her genes. In summary, we ran 7 experiments, and we found that in the cases where Concetta was most significantly different from normal subjects were cases at the lower luminance levels in our study. We see this as a sort of a sweet spot in the luminance scale where her differences in color perception are most apparent. So returning again to the questions that we asked earlier in this presentation, is it the case that independent of art training, photopigment genotype alone differentiates subjects tested in this task? Our tentative conclusion, given this preliminary data, is that it is not clear yet whether or not photopigment alone differentiates these subjects, and that more data is needed. The reason for this is because some of the tests that we designed were specifically set up to highlight the differences that Concetta might see, as opposed to general differences. The second question we asked was, does a life history of art training by itself seem like an important factor that is more important than photopigment genotype for this task? And the answer to this question, given this preliminary data, is no, it does not seem that art training alone can distinguish two subjects' isolumminate settings from a set of subjects that do not have art training. So further study is needed for this question as well. However, if we look at the second formulation of the question and we ask, does the life history of art training seem like an important factor in addition to photopigment genotype for this task? We can make the tentative conclusion that yes, indeed, although further study is needed, it seems like the conjoined features of having art training as well as photopigment opsin genotype makes Concetta Antico's isoluminate settings different from the three other subjects that we tested in this task. Finally, there are some issues worth noting about this particular study. It is important to realize that we do not actually know how Concetta's 4 cone classes are distributed in her retinas. We did not take measurements of her cone mosaic, so we don't have this information available to us. And it turns out that human retinas consist of a mosaic of cell types that is similar to the spotted pattern that you see in a calico cat, in that some patches express the pigment from the paternal X chromosome and the other patches express some pigment from the maternal X chromosome. This in human retinas has been shown to create highly variable photoreceptor mosaics. So some individuals, examples shown on the left, have photoreceptor mosaics that are highly M cone dominant, whereas other individuals have photoreceptor mosaics that are highly L cone dominant, as shown on the right. And we know that these kinds of variations in photoreceptor mosaic affects color perception. This is not the kind of information we presently have available to us about Concetta's retinas. The second issue worth noting is that we have not investigated any consequences of possible mutations that Concetta may have in her S-cone photopigment gene. Nor have we considered the contributions of other possible shifts that may be present in the L and the M cone photopigments on Concetta's photopigment opsin gene arrays. So these are still issues that could change the results that we presented here today and make them more interesting. Finally, I just want to mention that from Concetta's results, we've tentatively concluded that Concetta's observed settings differ markedly in some regions of visible color space from that found for the normal controls we tested. Specifically, the observed regions of Concetta's greatest deviations include stimuli involving substantial reddish spectral components, and this is consistent with the idea that her additional class of L' cones is driving the difference found in her settings compared to those of trichromatic controls. Secondly, in addition to the usual signals arising from the S and the M and the L cone classes that normal trichromats have, Concetta seems to have an additional cone class population in her retinas. We presume this to be the L cone pigment that is contributing to the luminance signals that are used in this task. According to these findings, then, Concetta appears to be using her potential for tetrachromacy in unique ways to reliably and significantly perceive brightness differences in some regions of color space where normals do not. Thus, results from this task suggest by using her tetrachromatic potential to evaluate reflectance spectra in particular regions of visible color space, Concetta differs from normal controls at a crucial early visual processing step, contributing to subjective color experience. The possible implications of these results, extending to the use of color relations in artistic representation, are interesting. One implication is that Concetta may have different color complements than the usual complements that are used in artistic representation. She may have different forms of color balance due to additional percepts that occur in her space. And we believe that perhaps in the representation of color in low light conditions, such as shadows and twilight and other low light viewing circumstances, Concetta may perceive and depict and implement in her painting a different degree of colorization than is seen in a trichromat artist. We'd like to just point out that in these studies we've used a completely new method to demonstrate some of the consequences of possessing a tetrachromat retina. Before this study, no one has shown these kinds of differences in a potential tetrachromat, let alone a tetrachromat artist. Many individuals will have the genetic potential for more than 3 photoreceptor classes, but we believe that few of these will exhibit strong perceptual differences from an extra cone class, because the optimal developmental environment for tuning up this capacity of tetrachromacy is rare. We are the first to argue and investigate through Concetta how, given the right genotype, early childhood learning experiences can create what you might call a perfect storm for realizing tetrachromat processing. We plan to further study Concetta's perceptual differences of light and color and hope you'll stay tuned for further results on her tests. The relevant citation for this talk is here. Thank you for your attention and for listening. If you're interested, you can find papers describing the details of this research elsewhere, as well as reviews of tetrachromacy in general. We would like to thank our colleagues at UC Irvine, Charlie Chubb and Christian Herrera, who provided assistance, space, equipment, and computer algorithms, as well as helpful advice in these scientific studies. Please note that the views and opinions expressed in this work are those of the authors and do not necessarily reflect the official policy or position of any agency of the University of California or the University of Reno, Nevada. The contents of this report are made public with express permission of Concetta Antico. You may cite this work in this way. And this work is licensed to Kimberly Jameson and Alissa Winkler under Creative Commons licensing, non-commercial, non-derivatives, and attribution. You may reach the authors by email, and of course your comments and questions are welcome.